Get started. Good. So today is the 13th lecture of our bioorganic chemistry course, and we have got to the last three lectures that I suppose is I hope is a good idea to devote to the neurobiology and neurobiochemistry. So these three lectures will be devoted basically to our different senses and how that looks like from the point of view of biochemistry and from the point of view of neurobiology. So today's lecture we start talking about chemical senses. That's basically olfaction, so what you smell with your nose, and taste, so what you can taste if you are trying some food or something like that. So regarding chemical senses, you know that we all have evolved in sort of sea of chemicals, and from the earliest organisms, some chemical cues, they signaled for us where is food, where is danger, and so on. So that was some sort of crucial mechanism for us, and this mechanism we have kept for all our evolutionary history, and even now they are still present in form of taste and olfaction. So this is the oldest and most universal sensory system. So even bacteria, they detect some chemicals. The interesting thing for the multicellular life is that we need not only detect some chemicals in the environment where we live, but of course we need to detect something in the internal let's say internal environment that we have. We have already considered in previous lectures with you the detection of, let's say, pH, the detection of levels of carbon dioxide. So we need that to know, do we need to breathe right now or so on. So that may be pretty crucial for us. That's why we have chemoreceptors throughout all our body. So we can detect something with our skin. For example, we can have some irritations, or we can detect some irritants on our skin or mucous membranes. Of course, in the digestive tract. We detect the levels of CO2 and O2 in some sense with carotid bodies. So we have considered that in one of our previous lectures. And of course, we should detect somehow the acidity. So you remember the Cori cycle and other biochemical pathways that leads to some sort of acidification when we are actively using our muscles. The interesting thing is that all our senses, they are in some sense, they are integrated together. I will show you a bit later in our subsequent lectures. I will show you how, let's say, hearing and seeing something will be integrated into some view of the world, and even seeing and understanding the dimensionality of the world and perspective is still integrated into some sort of general picture of the environment. But since today we are focused on the chemical senses, I should say that olfaction and taste, they are integrated into one thing, and we call that often flavor. So when we say that something has flavor, we often think about taste and smell combined together. And what's interesting regarding these sort of flavors, they are strongly connected to some sort of basic drives, including memory. So one of the best, the most memorizable things is taste or smell of something. So that may remind you of something you think that you have totally forgotten. But these things can get it out of your memory. So you can easily forget, I don't know, the Krebs cycle, but if you had at somewhere, I don't know, some bad meal and it tasted awful, you will definitely remember that maybe for the rest of your life. So it has really strong bonding, strong connection with our memory. What's more interesting, since all these signals they are integrated together, um, it's it may lead to some interesting effects. For example, there is a pretty interesting effect that if you, I don't know, somehow close your nose and you try to taste, let's say, onion and an apple, they will taste almost the same. I should not tell you that they are indistinguishable, because they are. They actually they are. You will still be pretty able to distinguish between them. But if you try this experiment, you will be, I guess, amazed how similar they are by taste. If you don't smell them, you just taste them. I think that will be a pretty surprise for you how similar they are. Yes, I guess we will have the second part of this lecture tomorrow. If you have some time and willingness, you may try at home tasting some onion and some apple, so just small pieces, but you should close your nose and try them. And tomorrow we may collect statistics whether that was, I don't know, whether they were testing differently, and were you able to distinguish between them and how similar they are. So did they become more similar than when you have feeling of when you had smell, let's say, activated. Good. So with that being said, we need to get a little bit into the neurobiology. So probably I should show you something regarding the anatomy of the brain. In many aspects, just some sort of a reminder of what it is and how that looks like. So here you have different mammalian brains on the right, including the human's brain and all the others. They are not drawn to scale, but for comparison, here you have strip on the left. So here it is. Here they are appropriately scaled so that you can compare the relative sizes of different brains. And what else I have here for you? I have two links for some sort of atlases. I'll be honest, I wasn't able to find some good brain atlas, some good 3D brain atlas. So these are the best I was able to find for you. So maybe if you can find something better, please share. I'll be happy to integrate that into the course. Good. I guess it's still loading. Oops. That's good. I think I have lost a part of Zoom. Okay, let it be. I have one more here, one more link here. That's Alan's brain map. So let me open it here. So what's interesting here, you have 3D views of the brain of a mouse. So here is a mouse's brain. You can look at different sections. You can look at views in 3D. And here you have adult human brain. There should be 3D somewhere as well. Where is it? No? Really strange. There should be some 3D for human brain as well. Maybe this one. I'm not sure. Okay. our brain fully loaded. So here we have it. This is a very simplified atlas, so not really special, but you can at least pose the mouse over some part of the brain, and you have certain description. So you see the isolate. Here we have it. So brain and different parts of the brain. Good. So maybe I will switch between images and 3D depending on what we are considering. So here you see once again the brain, and here you see different lobes of the brain. They are colored with different colors, and you see the appropriate names. So here is the frontal lobe, here is the parietal lobe, occipital lobe, and temporal lobe. So here it is. Here's cerebellum, and so on. Here you might see the central sulcus. So here it is, which splits the frontal lobe from the parietal lobe. And you can use that for orientation because we will have certain interesting structure in the vicinity of that, and that's pretty convenient. So here are different views of the brain from the above and from the below, or as you would say in the anatomy class, the dorsal and the ventral views. Here is the dorsal view, so here is the ventral view. Here you see the longitudinal fissure, so here is the longitudinal fissure. Here it is. And on the previous slide you had the central sulcus, and here you may find that central sulcus. Good. Here are some more views. That's view from the side, and here you see the directions like interior and posterior directions. You probably guess how the thing is oriented, and here you have some sort of slice. So you have a slice along along this longitudinal fissure, and here's how that looks like. So here you see the corpus callosum and all the different structures of the brain. So maybe I will show you some of them during this lecture. So here you see interesting structures. So here is the cerebrum, cerebellum, and here is what's interesting to us, the olfactory bulb. So here is the olfactory bulb in the human's brain. I guess I will have some better image later. If not, we will look at 3D. So the brain inside has these sort of ventricles. They are, let's say, I don't want to say the word empty because they are filled with the cerebrospinal fluid, or CSF. So they are filled with that sort of cerebrospinal fluid, and here you see that this fluid is basically synthesized by the choroid plexus, something around 500 milliliters per day. And that everything is connected to the spine, to the spinal cord, and here you see all that connections. So here are the lateral ventricles, here is the third ventricle, here is the fourth ventricle, and here is the aqueduct that connects the third ventricle with the fourth ventricle. I guess we have that in in the 3D view. So let me. So here we have open that in 3D. Here you see these verticals. Let me move that a little bit around so you may get some view of what's happening there. So here it is. Other structures we have. Let's check this. That's the end our what basically in here. So I guess good. Here is a view from the cross section. So here you see once again the third ventricle. The lateral ventricles are not seen, but you saw in 3D where they are. So you guess, I guess you understand the idea. So here is the cerebral aqueduct. Here is the fourth ventricle, and here is the spinal canal. So that's how it looks like. Here is once again view of the ventricles. That's some sort of projection. So you might guess how that looks. Good. Here's once again you have different lobes. They are all in different colors, and here you see the insula. So here, if we take this, the temporal lobe, if we move it away from parietal and frontal lobe, you will see the insula inside the brain. So here are some more terms, not for you to remember, but just to orient that how that looks. So here is the central sulcus. So here you see it, and here you see two gyri. One is the precentral gyrus, so here it is, and one is the postcentral gyrus, so here it is. Maybe some more interesting things like lateral or Sylvian fissure, so that's easy to identify and to spot on any brain atlas or image, and the superior temporal gyrus, so here it is. And here are some parts of the brain that are primarily processing some inputs, some sensory inputs. So here you see the somatic sensory input, here you see the motor cortex. So I guess I have shown you that video with the monkey from and the neural link project, and here you might see and I have probably told you that they have inserted the electrodes into the motor cortex. So here you see the motor cortex, and now you might guess where they were inserted. In the next lectures, we will consider some of the auditory pathways and visual pathways. So here you see something regarding that pathways. And here is the comparison of the human brain with the cat's brain. So here you see some large olfactory bulb. Once again, some visual processing areas, some auditory processing areas, and so on. And here is the comparison to the rat. So once again, different input areas and how that looks like. Well, of course, they are not up to scale, as you might guess. Some more areas designated on the human's brain. So here you see the olfactory bulb. Here it is. Here you see some prefrontal cortex, some premotor area. So here it is. So these part, some supplementary motor areas and primary motor cortex. So here they are. Some somatosensory cortex, some other cortices. So just for you to know where that is. What will be probably interesting for us, this small portion, that's so-called gustatory cortex. So we will talk about taste. So you might guess that some neuronal pathways they will go into this thing. I just wasn't able to omit that and not show you this nice gentleman, and that's a homunculus. And what it shows us, so here we have our cortex that gets some sensory input. So let me find that. Oh my god, I'm not moving the right way. Sorry. So here you see the somatosensory cortex. So here it is shown in green. And you might guess that if we are taking the inputs from all of our body, the parts of the somatosensory cortex taking that inputs will have different size. And that size is not even nearly proportional to the parts of the body where it takes that input. So the parts here in brain will be projected in the larger areas if we use them for some, I don't know, some tasks that involve very precise movements, so some precision is needed. So you might guess if we do many things with our hands, so probably hands should be projected into some sort of large area. And here you see that hands in this homunculus are pretty large, and here they are projected, so a hand, a finger, some fingers. So you may even somehow remap to the previous picture what we have in here and guess where everything is. And so you might guess we have a lot of cortex that is devoted to processing our face. So that's why touching our face, that's involves a lot of different neurons, and of course we need some sort of precision to move different parts of our face to, I don't know, express some emotions and so on. Here you see a large part devoted to hands because we are performing different precise movements and we need to use different tools. Basically we have evolved using different tools, and that's maybe even one of the definitions of the intelligence or some or something like that, the ability to use different tools or create different tools. So here is just a little bit more of anatomy. We are almost done with that, I guess, just to show you some nice pictures. So here you see the thalamus, here you see the hypothalamus. You might remember that part from our lecture that had to do with hormones. So we were considering different signaling pathways, and probably I should have mentioned that here is the pineal body, has to do with hormones as well. Here is the cerebellum. Here you see the tectum and tegmentum, pons, medulla, and so on. So you might guess that inside we have here the third ventricle, we have fourth ventricle, and so on. We have aqueduct. So that's it. Here you see once again the same view, but different parts are colored. So here you see the cingulate gyrus, the corpus callosum, the fornix, and so on. Here is the optic chiasm. I guess we will get to that at some point because I will show you the processing of the visual signals, and basically everything that goes from eyes, it gets to that optic chiasm and then to different parts of the brain, so I will show you that later. Here you see the hippocampus. You may have heard this term if you have ever interested in the memory and something like that, in how do we remember some things. so that's it. And here you see the amygdala. It has to do with some sort of emotion control, but I will not get into details, but certain let's say lesions or some defects in amygdala, they may lead to certain visible problems with that. Here's once again a view of the brain. So here we look from the upside down, so like this. So here you see the olfactory bulbs. We will get to them just in a moment. And here is the optic chiasm that we have seen, the optic nerve that comes from eyes, and here is the optic tract. And here you see the other different other parts of the brain. And here we have cranial nerves. I guess we need few of them, as they are innervating our tongue, for example, and here you see other parts of the brain. And just two cross sections, because I guess we will see these cross sections just a moment later. So here you see cross section of the brain. What's interesting, so here you see the brain, some side view, so so cross section like half of the brain, and here these number 2 and this line, they show how we have sliced this brain. So we have like taken some slice from the brain, and here we are looking at that slice, and here you see different structures. So here's some part of hypothalamus and so on. So here is insula. So you might guess if we move this part up, this a little bit down, the insula will be visible. Here you see the lateral ventricles, some parts of them, and so on. So here you see these parts. And here is another slice. It's taken from here. So here you see where it was taken from. And here you see all the different parts, so like fourth ventricle and so on and so forth. And here we have some parts of the pathway for the gestation that we need to consider today. Okay, so we have more or less went through some sort of anatomy of the brain. So we just dashed through it. Of course, I don't remember all that stuff. I don't expect you to remember all that stuff. That's pretty okay. But at least it's good if you can orient in that sort of atlases and find some structures if you need them, and orient in books that draw all these structures. Probably I will even close one of them that my computer freeze a little bit. Maybe I will show you another one, like adult mouse 3D viewer, so just for you to get the idea how that looks like. And then I will close them completely. So here's what we see when we start. So first, nothing interesting here, but we need to turn on view of anything. So let me turn on the view of cerebrum. So here it is, the brain of the mouse. Let me remove the reference grid. I don't think I need it. So here it is. What's good here, you can, let's say, search for some parts of the brain. So let me search for some sort of olfactory areas. So here they are. A large minus of the thing, it cannot represent adequately few different structures at a time. So when I'm turning on some structure, it is either obscured by a different structure. So even if I turn on some transparency, that doesn't really help. And you need to turn off some structures to be able to see, let's say, another structure. So here we see some olfactory pathways, but I can't turn on them both. So that's sort of minus of these atlas. Okay, but you should know that that we have these sort of tools. Okay, I guess I have lost some part of Zoom. I'm sorry, I maybe turn this on and off. No, did not really help. Okay, let it be this way. Okay, so part of the Zoom was lost, and I don't actually see some windows. But okay. Good. So here we have the taste. Of course, humans have evolved as omnivores, and we have a pretty flexible taste system to detect different things that we need. So probably we need some way to distinguish between what is safe to eat and what is not safe to eat. So that's why we have pretty complex system of taste. And of course you might guess that we have some sort of taste preference to sweet, and we have some rejection of bitter. So most cases something bitter, that will be some plant toxins. So you know that throughout our history we have relied for some time on some plant food, and we still need it as omnivores. So it's really that's a sign so that food may be poisonous and we don't need to eat it. We have let's say it's described some sort of 5 basic tastes. You should probably know them, like sweet, salty, sour, bitter, and umami. There may be some tastes behind these 5. I will show you that at the next slide. They are not described so well, but that's but we suppose that they are. What's interesting, I will show you in details some sort of biochemical pathways for all these tastes, but just to mention right now that anything sour, that means that you are tasting something acidic. And when I will show you that pathway basically detects the hydrogen ions or protons. So a really interesting moment. You cannot see a proton, you cannot hear a proton, so by no means, but you can taste a proton, and you definitely know how that tastes. So that's pretty interesting. Of course, salts, they are mostly salty. Many sugars, they taste sweet, and many proteins may taste sweet, and there are some artificial sweeteners that are basically proteins. The artificial sweeteners can be 10,000 to 100,000 times sweeter than sucrose, so we definitely have some ways to artificially hack our receptors and make them consider something as very, very sweet. Some bitter substances, they include some ions like potassium, magnesium, and some organic molecules. So most cases we are meeting with some organic molecules like quinones, and the thing that I have already told you, that we have the complex way of perceiving the food. That's the flavor that includes both taste, smell, and maybe even some other sensory inputs. And once again, you may test at home if you wish. That probably, if you, for example, exclude somehow the olfaction, you, for example, close your nose, the apple and the onions, they start tasting pretty similar. What's more interesting, some chemicals, like let's say capsaicin, that we have in that in that peppers, like red hot pepper, they influence not the taste receptors but the temperature receptors. So it really makes us experience some sort of high temperature when there is none. So that's interesting thing to notice, that the thing that we consider taste is basically not a taste, but let's say hacking our thermoreceptors. Good. Here is once again a little bit of anatomy, but this time not brain, but our mouth, throat, and nasal passages. So taste is of course a primarily function of a tongue. So here you see our tongue. Here it is. Here you see other parts, other interesting parts like palate, like nasal cavity, like pharynx, like epiglottis. Here it is. And the thing is that there are regions of pharynx, palate, and epiglottis that also have some sensitivity. Of course, largely tongue, but they have some sensitivity as well. Interesting that nasal passages are located so that odors from ingested food can enter through the nose or the pharynx. So some sort of retro-nasal pathway. So if you have even eaten something without smelling it, you can still feel some smell if it gets through this pathway. Good. So here is our tongue, and here you see the papillae and the taste buds. So here is the anatomy of what is happening in here. So papillae are the taste sensitive structure, and the largest and most posterior are the velate papillae. They have different forms. Let me find you. I guess here is a little bit better, maybe a little bit better slide that shows you different variations of papillae, and exactly that's three types, like circumvallate, foliate, and fungiform. So here you see the fungiform, they are mostly at the end of the tongue. Here you see the the here you see the foliate and the circumvallate. So here they all are. They have a little bit different shapes and forms, but despite that, they all have these structures. They all have similar. So here you see the cross section of a velate papilla, showing the location of so-called taste buds. So these are taste buds. In the next slide, you might see these taste buds in different forms of the papillae. So here they are on the fungiform, here's on the foliate, and here in the circumvallate. So here how they look like, and here is one taste bud in the cross section. And here on the right, you see the electron micrograph of a taste bud. So here how that looks. Let me return here a little bit. So here is once again a scheme. So here you see a taste pore, So you might guess that different chemicals, they diffuse to these through this taste pore, and they get to the taste cells that will be basically performing that sort of tasting. So so that's the taste bud is basically a cluster of taste cells, or the receptor cells, and the gustatory afferent axons and their synapses with taste cells and basal cells. So there is some sort of contact to the neurons. I will show you that in the following slides. And the microvilli at the apical end of the taste cells, they extend into the taste pore, the site where chemicals dissolved in saliva, they can interact directly with the taste cells. So here you have a lot of microvilli, and these microvilli, they will interact with the different chemicals. So each taste bud, it contains 50 to 150 elongated taste receptor cells. So in one bud you have a lot of different cells. They can react to different chemicals, as well as you have some supporting cells and the population of basal stem cells. So we are having some stem cells there to perform some regeneration, and basically tongue is, I guess, the best regenerating part of the human body. So yeah, if anything can more or less regenerate that some parts of tongue, so that's the best what we have. Pretty lame comparing to some different species, but we have what we have. Good. That's not so interesting. I have already mentioned you that besides the five main tastes like sweet, sour, salty, bitter, and umami, there are some thoughts that we may have some more. So we may have some more. Probably we have some sort of fat taste. We know that fat is calorically dense, and so it has some strong evolutionary value to detect that. And there is certain controversial because we can detect that by touch, so and texture, and of course that's not taste, it's just texture. But there are some evidence that like mice, they prefer fatty acid spiked water. So we suppose that maybe there are some fat sensitive taste cells, and there likely exists some sort of receptor for that. We don't know, as well as for the moment I was checking this, we didn't know the exact answer. So do we, or do we not have this type of structure? But maybe we do have. Maybe there is some sort of starch taste. So starch, of course, is some sort of glucose polymer, but it's not the question of the glucose detection. For example, if you are eating bread, you're not feeling that bread is sweet. So despite the fact that there are some large polymers that include glucose, you don't feel the sweetness when you are eating bread. If you keep that bread for some time in your mouth, for I don't know, maybe 15 minutes, maybe more, you will start tasting some sweet, because the alpha amylase that we have in our saliva will start degrading that sort of starch, and you will start detecting the separate units of sugars. But you can't detect that when it's in this form. But probably there is some preference in mice, so mice without some specific receptor like T1R3, they lose the sugar preference, but still prefer starch. So maybe there are some receptors that are capable of detecting that. We don't know, but maybe there is. There are some other tastes that we may describe as humans, but probably there are no specific receptors for that. Like carbonation taste, that's probably the result of somatosensory tingle. That's when you are drinking some sort of fizzy water, and that's the taste you get. Some calcium taste, that's some sort of bitter plus sour, and calcium taste requires T1R3 in mice, while in humans it is reduced by T1R3 blockers. So probably there is some version of calcium specific taste receptor. And of course, we all describe water as having some water taste, but there is no water taste receptor identified yet. So probably we don't have any. While insects, they have some clear water receptors. So that's good. Moving further to the more detailed biochemical view. Here the taste bud and some cells from that taste bud are showing different recordings when they are under the influence of different chemicals. And here you see some gustatory afferent axons that are going from these cells. Well, I will show you these sort of junctions in a little bit more details just in a few moments, but let's look at some sort of general picture that we have. So here we have some some general picture. Here you see different chemicals like sodium chloride and quinine and hydrochloric acid and sucrose, and we are acting on these different cells. Of course, we expect them to detect some sort of saltiness, bitterness, sourness, and sweetness, depending on the chemical that we are using. They have different sensitivity to that, and in the case the action potential discharge of the secondary axons, here they are recorded, and you see the firing of the neurons depending on what we are tasting and how the cell reacts. And here you see the number of action potentials, let's say in the span of 5 seconds. And here you see that number, how these cells react to different chemicals. Okay, so what's interesting? So the chemically sensitive region, that's apical end with the microvilli that is projecting into the taste pore. So you have seen the taste pore on the previous slides. So this is projecting into the taste pore, and this is the taste pore itself. So here you see that in the image, and here it is in the micrograph. So here they are. And basically here they are in the image of the tongue. So I hope I have positioned ourselves as precise as possible where we are in the organism. Taste receptor cells are not neurons. Interestingly, when we get to olfaction, the cells that are detecting different smells, they are neurons. These are not. But still, they have synapses with gustatory afferent axons, and the information through the axons will be forwarded somewhere into the brain. So they form electrical and chemical synapses with basal cells. So I guess I will show you that in the next picture. They constantly renew, so they live like approximately for 2 weeks, and you might guess that's why we need that sort of basal basal cells to renew the taste cells. There are different ways to transmit signal. Sour and salty are transmitted through releasing serotonin, and sweet, bitter, and umami, they are the ATP is released. There may be some other transmitters present, but their role is unclear. So I will show you exactly how that works on the biochemical level in the next slides. Just a few points to notice. Most taste receptor cells respond primarily to one basic taste. Some taste cells and many gustatory axons are broadly tuned, and response patterns depend on specific transduction mechanism expressed in each cell. So we will get to that mechanisms, but here you see on the previous image you have certain taste and you have cell responding mostly to that one taste. Good. Now let's get a little bit into the biochemistry. So here you see the saltiness detection. There are basically two pathways. One for the low salt detection, that's shown here on the right. The other pathway is for the high salt detection. So if you have a very, very high salt concentration, that will taste really bad and aversive, and that activates bitter and sour taste cells. So to cause the avoidance, because we don't want a lot of salt in our organism. The exact mechanism is still unclear, so I can't explain that because we still don't know. But we know that different receptors will be activated. So what we have here on the right. So the salt taste, that's primarily the taste of sodium ions. So here you see sodium ions, and they are getting into the cell through the amyloid sensitive sodium channel. So here you see the ion that gets in. So they are always open. These channels are always open, and they are voltage insensitive. So they are different from the channels that we have considered in our lecture on action potential propagation or something like that. So what happens? The sodium diffuses inward. That causes the depolarization. So sodium gets inside, and that depolarizes the cell membrane. The cell membrane depolarizes, and due to this depolarization, now the voltage gated channels are open. So here we have voltage gated channels. So this one is not voltage gated. These two, they are voltage gated. So they are basically reacting to the fact that we have some depolarization due to sodium getting inside. So when the depolarization gets to some threshold, they open. That leads to more sodium getting inside, more calcium getting inside, and here we start the process of releasing neurotransmitter. So here we have synaptic vesicles that are filled in with the serotonin. So here is serotonin packed inside, and here we start the process of releasing the neurotransmitter from that vesicle. The process, I'm not getting into the exact details of this process because we have already considered that. So we have already considered the mechanism of calcium-related release of the neurotransmitter when we were considering the neural terminals and releasing of the neurotransmitter there. So pretty the same process we have in here. And here you have the gustatory afferent axon. It can detect this sort of serotonin, and it will transmit information to the brain. So, brain, we have something salty in our mouth. So acts appropriately. So if we need that saltiness, let's eat it. If we don't need it, let's not eat it. What's interesting in here, there is some sort of potassium preserving diuretic called amiloride, and it can block these sort of sodium channels. So here's a tricky question for you. Would you predict how our experience of eating something salty will change in case, I don't know, I have performed some mouthwash with the amiloride? Probably not worth doing that, but it will. Yes, it will change. How it will change? With amiloride, that's the chemical that basically blocks this channel. So this channel will be blocked. It is used as some sort of diuretic that is potassium preserving, but it has this sort of yes, if you have some low levels of salt, the whatever you have eaten will not feel salty to you, but that up to some point, because since we are blocking these channels, sodium cannot get inside, so we cannot depolarize the membrane. These channels are not opening. These synaptic vesicles are not releasing the neurotransmitter, so no signal, no action, no smell. Well, we are talking about taste right now, so smell will be in the next lecture. Good. Yeah, I guess I have lost somehow the side panel of the Zoom, so I'm not seeing you completely. Oh god. I'm not sure how can I change that so I can open everything, but I cannot. Better, but not so good. Oh. Or I have broken everything. I have broken everything. I'm sorry. How do I fix that? Okay. Okay, that's why we don't like all that programming things. Good. So we still have some time, so let's get through other tastes. Here we have soreness. So what is soreness? That basically signalizes you that we have low pH. So you, I guess, remember that pH is minus logarithm of the concentration of the hydrogen ions or protons. So the high acidity means you have a lot of protons, but since you take minus of logarithm, you get some low numbers. So some acids, they release these sort of protons, and they are the primarily stimulus for these cells. So as I told you, you don't know how the protons look like, but you know how they taste. So now what's happening in here? Pretty interesting mechanism. So here you have a potassium channel. So this potassium channel has some sort of permeability. So potassium can get out. When we have protons in here, they are blocking this potassium channel. Moreover, they are doing that from the both sides. So they are blocking. They are blocking it. They can either get into the inside of the cell through the proton channel, or they can remain on the outside of the cell, but still they can block the potassium channel. If the permeability to the potassium is decreased, you might guess that the membrane depolarizes. So getting outside, potassium repolarizes the membrane. So I remind you from our previous lectures, so we had some sort of action potential. We were considering an action potential, so you remember this form of the action potential. So the way down, that was potassium getting out. So we have depolarized the membrane, so sodium has depolarized the membrane up to some point, and then potassium channels were opened, and we were repolarizing the membrane back. So if we have some continuous flow of the potassium, it is trying to repolarize the membrane and increase that sort of potential difference that we have between the outside and the inside. If we are blocking these channels, we are basically stopping that mechanism that helps us to make the difference bigger. So the difference gets smaller. The smaller difference means the depolarization of the membrane. We have some depolarization. Once again, we have channels opening. So we have sodium getting inside and further depolarizing. We have calcium getting inside. And that, of course, triggers the mechanism of the serotonin release. So once again, we have some vesicles, and here we have the release of the serotonin. Good. So there are likely multiple mechanisms together that create perception of soreness, but at least this one is more or less well known. So here's what we have. Now we get to a little bit different receptors. So here are receptors for bitterness. So these are receptor families, the whole receptor families. Interesting thing is that bitter, sweet, and umami detection uses T1R and T2R receptors. They all are G protein coupled receptors, so GPCRs. So once again, we return to our G proteins. Here you see that sort of receptor in the image. So this is a GPCR. Interestingly, they are all dimers. So if receptor is functional, that's a dimer. So that's why you see in the image two of the receptors. Here you see different bitter receptors, the T2Rs. Humans have something like 25 T2R receptor types. The main purpose is some sort of poison detection. So if something tastes bitter, it's probably some sort of poison. So that's why this taste may cause so much aversion. So we don't like it. It's probably something poisonous and better not eat it. So it's a danger signal. Here you see these sort of receptors, and here let's consider the mechanism. Pretty typical for the G protein coupled proteins, and for the G protein coupled receptors. So here we have some GPCRs. They interact with the chemical on the outside. If that's some sort of right chemical, so we are detecting that this chemical should induce signal. we have G protein activated. I hope you remember all that mechanism with G protein being constructed of three different parts, and these different one of that parts goes through the exchange GDP to GTP. It's activated. It then can move somewhere and let's say activate the phospholipase C. So here we have phospholipase C. This phospholipase C is capable of making inositol 3-phosphate. It takes phosphoenol pyruvate and makes inositol 3-phosphate, and that inositol 3-phosphate basically triggers calcium release from some sort of calcium stores in the endoplasmic reticulum. We have seen similar processes in different cells as well. If you recall, for example, our lecture, I don't know, on muscles, they were heavily relying on the release of calcium from the sarcoplasmic reticulum. Here we have some sort of reticulum and calcium release as well, but now triggered with inositol 3-phosphate. So here we have this calcium, and now these calcium and inositol 3-phosphate, they are acting on the sodium ion channel called TRPM5. So here is TRPM5. This channel, that's some sort like two keys that you should turn on. So some sort of additional precaution that you need two signals to turn on this sort of channel. And that further depolarizes the cell membrane, and this depolarization together with calcium opens ATP permeable channel. So this channel will be opened if you have both depolarization and you have calcium. That will release ATP from the cell. That ATP will be detected with the gustatory afferent axon, and the brain will receive some information that we have something bitter in here, so maybe we should avoid that. Here are sweet receptors. You might guess they look pretty similar, so that's dimer once again. On the previous slide we had T2Rs, two dimers of T2R type receptors. Here we have one T1R2 and one T1R3. So here's once again some dimer, and this is once again a G protein coupled receptor. Pretty similar mechanism. So the same mechanism inside. So we have once again the phospholipase, all that inositol 3-phosphate synthesis, so on and so forth, up to releasing ATP. Once again, you have ion channels TRPM5. So many chemically diverse sweet molecules, they are all detected by the same receptor type. This is the sweet receptor T1R2 plus T1R3, so that's a GPCR dimer, so we definitely know what it is. If either subunit is missing, we cannot detect anything sweet, so no sweetness. Interesting example, cats, for example, they lack functional T1R2, so they cannot taste anything sweet. So that's maybe like fun and useless fact for you. So cats cannot taste sweet things, and that's because they lack T1R2 receptor. And you might have noticed that inside the cell, after the signal gets to the GPCR, we have absolutely the same pathway as we had in the case of bitter taste detection. So why then sweet does not taste bitter? So why do we have two different tastes? And everything boils down to the fact of which kind of afferent axon we are using. So these axons, they know what are they connected to, and basically the fact that we are transmitting that information through some axon basically defines, do we feel something sweet or bitter or something like that. And here is just a fun video for you. So that's if it starts, of course. So that's my dog, and he's just craving sweets. I don't know. I need to do something to keep them away from him. And here is some sort of ice cream that's pure plombier, and he really likes that. So I gave him some small portion. Okay. Taste of umami. That's savory taste of amino acids, especially glutamate. So you might guess we need amino acids because we are we need them to construct our bodies. So of course we prefer some foods that is protein rich and nutritious. We can even gain some energy from them, so that's pretty good. The umami receptor, once again, GPCR dimer, this time T1R1 plus T1R3, so here it is. Absolutely the same intracellular biochemical pathway. GPCR, phospholipase 3, releasing calcium, letting sodium in, depolarizing the cell membrane plus calcium signal, releasing ATP, and the afferent axon sends an action potential somewhere to the brain, where it will be processed. What else? Very similar to the sweet receptor T1R2 plus T1R3. T1R3 is shared between sweet and umami. So T1R1 determines the amino acid sensitivity. So that's, I guess, pretty interesting biochemical fact. So you have dimer, and when you switch one part for another, you get pretty different sensitivity. So, and if you make mice knockout in T1R1 gene, I hope you remember how do we make knockout mice. I have definitely made some small portion of the lecture regarding CRISPR-Cas, for example, and these mice, they cannot taste glutamate. What's interesting that many bats, they lack functional T1R1, so they have no umami taste. Some vampire bats, they lack both sweet and umami receptors, and that's probably some sort of evolutionary feature, like they don't need that taste for survival. Now let's get, since we have still 15 more minutes, let's get to some more useless fun facts. Here you see so-called Syncephalum dulcificum. That's a plant of a Sapotaceae family native to tropical Africa. And it's known for its berry because when eaten it causes sour fruits such as lemons and limes subsequently consumed to taste sweet. And that's pretty interesting. So some common names, they include like miracle fruit or miracle berry and or sweet berry or something. So what the reason? The reason is so-called miraculin. So here you see this sort of protein, it's called miraculin, and that's a taste modifier. That's basically a glycoprotein that is extracted from the fruit, and it works by binding to the human sweet taste receptors and changes them from inactive state at neutral pH to an active sweet tasting state in the presence of an acid. So this thing binds to our sweet receptors, does nothing, but if pH drops, it changes conformation and activates that receptors. So that is possible. So you might guess that if we have the thing, if we have eaten something like this, or I don't know, at least perform some mouthwash, with that sort of thing. We will taste whatever is sour, we will taste as sweet. And here I guess I have two videos for you. So that's some pretty interesting science channel. And here you see this guy, so Denjiro Sensei. He's trying to eat lemons after he has eaten some of that miraculin. And here he's trying to do that in some sort of competition. So he will try to eat some lemons and compete with who can eat more.
今日は舌で感じる感覚、味覚がテーマです。中京テレビの伊藤アナに来てもらいました。よろしくお願いします。
お願いします。
伊藤さんはレモン好きですか？
はい、好きです。
では私とですね、どちらがレモンを早く食べれるか勝負です。
はい、頑張ります。
So these these are parts of lemons. So just lemon that was cut in slices.
準備はいいですか？
はい。
ではスタート。
So you see he's cunning, so he ate some of that miraculin and now he's just devouring that lemons. And here the poor girl is trying to eat them as they are, and you might guess they are pretty sour. I don't really like lemons on my own, so so I'm even feeling a little bit bad for her.
酸っぱい。先生、すごい。
はい、私の圧勝でした。実は私、科学的にズルをしていました。あらかじめこれを。
So here you see these berries of that contain the miraculin. So here's even a photo how they look like in the nature.
食べていました。ミラクルフルーツという果実です。これを口の中に入れ、舌の上で転がします。すると酸っぱいものが甘く感じられるようになるんです。なぜそうなるのか、簡単に味覚の仕組みを解説しましょう。味を感じる仕組みは鍵と鍵穴の関係に似ていると言われています。舌には味蕾という味を感じるセンサーがあって、これがいわば鍵穴。
So here you see that's one of our previous slides, basically.
です。例えば酸味成分は甘みを感じるセンサーにははまらず、酸味を感じるセンサーに鍵のようにはまります。その時に酸っぱさを感じます。ミラクルフルーツを食べた場合は、ミラクルフルーツに含まれるミラクリンが酸味と結合します。すると酸味のセンサーではなく甘みを感じるセンサーにはまるようになります。
So this says some sweet sensor or sweet detector, and these say some sour detector, and that's how he's explaining the action of the miraculin. So I would rather maybe say that this thing will sit in the sweet receptor and wait for something sour, and now we know that's a proton to come in and change the conformation, but
そのため酸っぱいものが甘く感じると言われているのです。では伊藤さんにも体験してもらいましょう。ではミラクルフルーツを口に入れて舌の上でまんべんなく転がしてください。ではそちらのレモンを食べてください。
うん。 甘いですね。
ミラクルフルーツは簡単に手に入るようになりました。ぜひ試してみてください。
ミラクルフルーツは糖分の制限が必要な方へ甘味剤として。
So now you see that after consuming this sort of fruit, well, the lemon tastes much better. Good. Let me check just for a second how many more slides do I have to consider. Well, that's almost everything. Great. I guess we are doing great today. Maybe except for the fact that I have broken my Zoom, but let it be. Good. So now we have considered the biochemistry of how do we get from some chemical to some signal. So all that pathways inside the cells, they are basically converting whatever we have into a signal that should go to the brain, and then the brain will interpret that somehow. But let's take a look now on the pathway that the signal should go through to get to the brain. The taste information from the tongue and mouth cavity is carried by 3 cranial nerves. Nerve number 8, 9, and 10. So here you see the image. Here you see the primary gustatory cortex, and we should somehow transfer our information from here to here. Here you see cranial nerves. There are 12 cranial nerves, and thanks God we're not studying medicine, otherwise we will need to remember them all by heart. So where do they go and what do they do? But we will not, and that's pretty good. In one of our previous slides, let me return back to the anatomy. So here we had all these cranial nerves, so you might get the idea where they originate from. So here they are. Now returning returning here. So you see these 3 nerves, they are innervating different parts of the tongue. So here is the cranial nerve number 8 innervating the end part of the tongue. The number 9 is innervating what's deeper inside, and number 10 is basically innervating the epiglottis. And they will all carry information to the brain regarding what we have eaten there. How do these nerves get in? So here you see these nerves. So here they are getting into the brain, and we will be interested probably with these slice. So here you see slice number 1. So here it is. So here's the slice number 1. So afferent from tongue and epiglottis are getting here, and here you see the left gustatory nucleus. Of course you have a symmetric thing on the right, so we're just considering one part. On the right you will have the same thing. The gustatory nucleus, once again I will return a little bit to our anatomic slide. So here is the same slice that we had. So if you would like to position some other structures with respect to that gustatory nucleus, so here you can look at this slide and see some different things. And the other slice that we will have will be this one, so this slice. So here it is, and here we will have some structures regarding the gustation and taste as well. So if you will reconsider after the lecture all the things, you can return to that slides and maybe see in more details what else is seen in that two slices. So gustatory axons, they enter the gustatory nucleus within the medulla. The gustatory nucleus axons synapse on neurons in the thalamus, which project to primary gustatory cortex in regions of the postcentral gyrus and insular cortex. So here you see that signal came here to the gustatory nucleus. Now this signal is transported to here. So here you have the left ventral posterior medial nucleus of the thalamus, or VPM, and now from this nucleus you have signal sent to the primary gustatory cortex. So here you see where the neurons end, so here some end in the insula, so here some end, and so on. So that's the way the information passes through to get to here, to our some sort of gustatory cortex, where it will be somehow interpreted and maybe transferred to different areas to be combined in the overall view of the world around us. And here you see at the bottom some sort of summarized taste pathways. So you see the different parts of the tongue. They send signals to the gustatory nucleus, to the VPM, and then to the gustatory cortex. So that's the pathway as we know it. Good. So that I have basically described to you. Some projections from gustatory nucleus to brainstem regions control swallowing, salivation, some maybe gag, vomit reflex, respiration, and digestion. And there may be some additional projections to the hypothalamus and amygdala that regulate food motivation and palatability, appetite. So lesions in these areas, they may cause overeating or food aversion or loss of interest in food. So there are some additional pathways, but anyway. Regarding the coding of the taste, so many receptor cells, they respond mostly to one taste, but partially to others. So mostly one, but maybe some small response to something else. Some primary taste axons, they are even less specific, so the central gustatory neurons in the brainstem, they are broadly responsive. And of course that begs the question, how can we distinguish between different tastes if something is broadly tuned? And the thing is that a single receptor cell may use multiple transduction mechanisms. So one gustatory axon, it may receive input from multiple taste cells, and gustatory nucleus neurons, they receive input from many axons. So you have some sort of overlapping response patterns, and the thing is that you could have made everything specific, so like send just one signal through one neuron and so on. But what would happen if we have certain troubles with that neuron? We just completely lose any sense of for some particular taste. That's no good. So we need some system that is more robust and less error prone. And as a result, in many cases, not just this case, but in many other cases, brain uses so-called population coding. So it does not code like one taste, one neuron, or something, some one unit of something and just one neuron that corresponds to that unit. It uses some patterns across many neurons, because that adds you much more stability to the system. If something fails, at least not everything fails, so at least something will continue functioning, and we will still get relevant information regarding the the environment. Before ending the lecture, I guess we are almost there. I still want you to show yes, just 2 slides. Maybe I will show you this one. I want to get a little bit into the genetics of the taste. As you might guess, we have some genes that encode all these receptors. And you can probably guess that if we have genes something, there will be some genetic variation of that. So it's really rare that you don't have any variation. That should be, I don't know, some hypercritical part of the biochemistry of the organism, that any change will just kill the organism. So many things are not so critical, and some variations are still possible. So of course the receptors still can be varied, and that means that we are inheriting some genes that define how do we taste something. For example, there are several TAS2R bitter receptor genes that make you more or less susceptible to the bitterness of the alcohol. It is thought that there are some bitter congeners in alcoholic beverages, and depending on that, you will taste it as more bitter or less bitter. Some genes like TAS1R1 and TAS1R3, they correspond to the variation in the perception of the savoriness. So these are variants that alter sensitivity to the glutamate. So here you see the glutamate in this image. Probably you have heard about sodium glutamate. It's added, I don't know, almost to all food that we have, especially anything that should taste like meat. So that's pretty common chemical used everywhere, and depending on the variations of the genes, you will taste that like more or less savory. But here is even some more interesting examples. We may have some sweetness sensitivity. Once again, genes TAS1R2 and TAS1R3, they like give you some differences in the perceived sweetness of and sugar preference, and these are variants that modify the sweet receptor. But probably the most neat thing that I want to show you, and that is often used in some sort of different demos across like different courses, that's the taste of bitterness, especially the gene TAS2R38 on the chromosome number 7. Depending on the variant of this gene, you may taste like incredibly bitter one of these chemicals, so phenylthiocarbamide, or here it is, so PTC, phenylthiocarbamide. You see how that looks like. Or you can use different chemical with the that does pretty the same. That's 6N propylthiouracil. So here it is. It's called prop. So here you see the formula. You might even guess where does the thia comes from. You guess which part resembles uracil and which part resembles what is propyl here. So I hope you remember all that parts from our chemical part of the course. So what's interesting, depending on the haplotype, you may have strong response to that. So you feel that incredibly bitter and really aversive, so very, very bad taste. You may have some less response, so you feel it bitter, but it's not so bad. So yeah. And you may have even no response, so you don't taste it at all. And that's pretty interesting part. Here I want to show you some sort of experiment. So here children are given some sort of I know some stripes that were soaked in one of these chemicals. I'm not sure which one of them. Earlier, everyone used the PTC. Now it's more common to use prop. It's thought to be better. I don't think we can get any of these. Probably everything is forbidden for us. But okay, let's get to the video.
3, go.
It's so bad!
God, doesn't taste like anything.
How many of you were non-tasters? How about tasters? How about super tasters?
Do you have the bitter taste gene?
So here you see how children differently react to the same substance. So some of them does not show anything, they just look like it's nothing, and some of them well show pretty they definitely tell you that it tastes really bad, so they don't like it. So once again, if we had ability to get any of these and do lectures in some more live form, so maybe we can make this sort of experiment, but not this time. Good. I guess with that being said, I have completed the today's lecture. Yes, questions. How does depolarization in the taste cell ligand binding at the taste receptor? Okay, let's return back to the slide. Second messenger, then this should be something from these pathways. Good. Yes, so here you see the I'm not sure if I fully understand the question, so so here we have a receptor, so let's say a bitter receptor or whatever. So here it is a G protein coupled receptor, so here we have G protein. This G protein it activates the phospholipase C, so that will generate the inositol 3-phosphate. So that's the second messenger, the second messenger cascade. This second messenger, together with calcium, activates TRPM5. So here it is. That's the channel that lets sodium come in, and that leads to the depolarization of the membrane. Now calcium, together with the depolarization, so probably I should draw here some, I don't know, some arrow, so depolarization plus calcium allows ATP to get out of the cell. So only calcium or only depolarization will be probably not enough, or it will not respond enough potent to activate some action potential in here. So you need both keys to be turned on so that you get enough ATP out of the cell that can interact with this sort of afferent axon. Yes, so we are just changing some sort of potential difference across the membrane, and you see that all these channels, they can somehow react to the changes in the membrane potential. Good. Any more questions, comments? Please fire away. Well, Maybe that's because of electrolysis that is starting on your tongue. So you have some water and you have some salts, and you know that if you put any battery into salty water, or well, even in the tap water, you will see some bubbles coming out and some electrolysis going on. So probably there's some sort of result of that electrolysis is acting. Well, I will double check before the next lecture, but my hypothesis is that you have a few different processes, including some sort of electrolysis, and the products of that electrolysis, they act on the sour receptors. Good. Any more questions, comments, please? Okay, I'm sorry, I can see the views. Good. Now I do. Okay, if no more questions or comments so far, then thank you for your attention and see you tomorrow. Have a nice evening.